want to thank the panelists for their view. Now we have an amazing set of talent here, and I want to open it up to the audience for questions. Yes, at the back.
If you were the Chinese government wanting to invest some of your large budgets, where would you invest it? In what areas would you invest? Assuming synthetic biology.
Mortgage banks.
Right.
Yeah, after you sell your Morgan Stanley stock. Reg and I traipsed around China with the view that there might be an opportunity to do something like the QB3 UCSF, Berkeley, Santa Cruz idea to accelerate the technology that did have commercial promise. And surprisingly enough, what we found in the specific field of synthetic biology in China was very, very, very far behind. And Reg could address, what is being done, but there's work in pharmaceuticals, there's work in, in various scientific fields, but the field of synthetic biology is way behind. So in fact, after meetings in Shanghai and Beijing with the leading academics, actually with venture capital groups as well, we decided it was just simply too soon, that it was going to have to be developed in the places that it's now being developed, and that a conference like this, for example, which has attracted scientists from China and investors from China, may in fact help to accelerate that. But it's one of the things that, to me, confirms that a place like Hong Kong, where the university science, in fact, is surprisingly ahead of China, presents a better Asian opportunity.
I have to say that at lunchtime, Jack suggested that China should use its money to buy America.
That's over. Leftover investment.
Another thing they could do, and one of the differences, when I moved out of academic labs and into industrial labs, and probably best epitomized, I'm sure it's probably like this at Amyris, when we formed Nanofluidics, which is now Pacific Biosciences. In the first 15 people, there were 3 computer scientists that we stole from Apple. There were 3 optical engineers from IBM Labs and from Optical Networks. There were 3 molecular biologists. The point is, if you in one room sharing the same cafeteria and working right next to each other, you have very, very disparate disciplines. And one of the best parts of QB3, if you go in there, and my friends who are in the physics department are growing yeast. And I think that, and similar, in the Bay Area, there's several centers in BioX. In universities, you formalize it with the building, and you make people sit next to each other and share space, and it's incredibly productive. So if you want to invest some money and do something that's sufficiently different that will almost always be valuable, you make people from very, very different disciplines work on the same problem. And the nice thing about companies is you're all working on the same product, the same project, and you all have the same stock options. It's a little more complicated in academics, but it can be done, and that's a great experiment. And synthetic biology is certainly something that's benefiting from having engineers and software guys doing biology, and that's really one of the attractions of it.
So I'd like to answer it a little bit differently and talk a little bit about what we're trying to do in the Bay Area. It's something that Drew has convinced me of, and I've sort of become an acolyte of his in trying to create a biofab in the Bay Area. And what shocked me when I began to get involved in biology are how primitive the tools are. So I'll give you an example from my experience from one of the companies that we invested in. They were doing a prototype project that they had 2 years and $2 million to engineer a yeast that could metabolize C5 sugars. And so what the first 2 months of the project, if you ask any of the VCs, there's only one word there's one word that we start every meeting with is fast, right? How fast can you do this? Can you do it faster? Please do it faster.
Oh yeah.
So the first 2 months of this project, they spent designing a vector, which is sort of like, I don't know, writing a text editor in order to start on your novel. And so the idea behind the biofab is to begin to build these basic tools, the hammers, the nails, all the pieces and standards that you need in order to effectively pull things off the shelf and do projects very much faster, much more capital efficiently, and to enable innovation. So if you were to ask me, I'd say put it in the project that I'm pushing, which is the biofab. But the biofab doesn't have to be in San Francisco, and we don't want only to have one in the world. We want it to be everywhere.
Go ahead.
Actually, I'd like to just follow on from that. Actually, maybe a question for Jay and Zach. Question for Jay and Zach, picking on you as the representatives of sort of a company already doing this. I mean, Drew and Tally have talked about the biofab, and a lot of people here have talked about trying to reduce transaction costs and IP costs with things like the BioBricks. I just wonder if you give your view on whether you think a lot of those things would reduce costs in your specific instance, or whether a lot of the cost that you're dealing with is actually the follow-on work from having designed the microbe, to actually develop the market for the fuels and stuff like that, and where you see the balance of those costs coming out.
Well, I certainly think the biofab would be fabulous. If you had a bunch of parts you could just order in that were well characterized, you could start to design and get something that would actually work. Maybe the first pass through or slightly work. That would even be a big improvement. So I'm a big proponent of the fab. Certainly you've got all that back end stuff, but the problem with biology, as everyone has alluded to at this meeting, is that it's very difficult to construct up front and construct it as and get it to work as you've designed it originally. So I think the fab is a great idea. I think we really need it.
Sure. This may be a bit of a technical answer for this session, but I talked to Drew after his presentation. I think the biofab is a great idea. I also know from having worked in the field for a while that S288 yeast and E. coli are unlikely to be very good host organisms for a lot of industrial applications. And so the chassis matters. And if you're going to be standardizing parts and studying their properties, the context in which you do it will also matter. I will also say that Amyris is already engaging in biofab-like processes with regards to part characterization in our own hosts that we are using. So we clearly see it has value, and we're not waiting for a public consortium to make it possible.
Zach, do you want to mention why it is you don't think bacteria are such a good platform or a good chassis?
They are in some applications, but at very large scales you have issues around phage and contamination. Competition is always going to be a problem. If you have to purify all your components that are going into the fermenter, that comes at a tremendous additional cost. And that cost, in biofuels at least, is prohibitive.
Back, yeah.
I would like to ask Jay that if so all these applications are or all the type of applications which are coming are kind of small type of applications. They are not like medical applications.
I'm a small thinker.
Well, I would like to think big. So if you have some idea and you really don't have the money to pursue it, but you are really confident about that idea, then how would you go about it? Like, because you initially you need some money and time to prove it as well, right? So then if nobody is willing to pursue it in the academic environment, then how are you gonna follow?
Well, so first thing is you want to get as much of the project done before you take these guys' money, right? Because the minute you start taking their money, as an entrepreneur, you get diluted. So you want to get as much of it done before that as possible. Nothing against that, but as an inventor, you want to get as much done as possible. So you write, if you're an academic doing this, you try and write to get grants from the government. The more you can develop it in an academic lab with grants, the better. Universities are often very proactive now in helping you work through some of those situations where you can maybe get a company started either within the lab or at least get the ideas, get a good foundation for all of the ideas before you move it out. So grants are a good way. These guys can talk about alternative funding much better than I can. I guess the majority of the things I've done, almost everything, has started with an idea that got funded through a federal grant.
One more thing I would like to ask. So do you publish on the results, or do you don't publish till you are finally convinced that this idea works and you have the money and so then how would you how would you protect it from
Well, a patent's about an idea, right? And so you file a patent. Of course, you have to reduce it to practice, or should, and you certainly need to do that if you're going to start. Well, a lot of companies have been started without reducing it to practice too. That's not such a great idea. But so what you want to do though is you want to protect the idea with a patent application before you publish on it. That's essential. But I guess what I was saying about patents and publishing is that these can be done together. As you're writing the paper up, you can write the patent up, and so that when the patent goes out, the paper can then go out.
Okay. Thank you.
On here, because this is a little bit my area, because I run an institute which tries to be between the academics and the venture capitalists. And one of the things that we've tried to do is raise funding. So we have about a couple of million a year to give away to people like you say. When you need to do bridging the gap funding, from an idea, which is too late for NIH funding, but it's too early for venture funding. We've tried to raise a particular fund to help our academics get over that gap. It's a very good point. Next question. Yeah.
Just want to hear comments on business models for open source biotechnology.
There are a few, and I think that whenever you see a new business model, you can either worry or you can get excited about it. Clearly IBM PCs in 1981 was an open architecture. Not every aspect of any open source is truly open. In other words, there's always some component somewhere in there that is closed, as far as I know. The open source software runs on an Intel chip, some of which is closed. We've run a pretty radical open source effort on sequencing, which is open source hardware for everything that we have any control over. So there are components in there, and we'll see how that turns out. But I think the IBM PC is one of the older examples. But some of the open source people may not accept that.
Yeah. So I graduated in '99, and then started doing right before the crash, and so basically my career started in the crash, and I started doing free software for nonprofits during a recession. So I have a pretty good idea of open source business models. I have a pretty good idea of open source business models, and I've tried to do it a number of times. And so my opinion is they're terrible. They're the worst things you could possibly do. But that doesn't mean people haven't been very successful. Guys like Red Hat have done it, and a number of other people. And they've wrapped the core with proprietary offerings and service offerings. But I don't think that that's necessarily where the open source biotechnology is where it'll be powerful. It's for Amyris and LS9, the next Amyris and the next LS9 companies we found and invest, that they can do projects faster. They can take cheap capital. So the issue is, do we really have to go to Invitrogen for every basic piece of genetic? Where are you from? So okay, I'm sorry. Yeah. So I'll see you outside. So anyway, the goal is to really try to make it cheaper for people, to put pressure on Invitrogen and give them a little bit more competition, and open the space up.
So it's probably important to note that not all companies should be funded by venture capital. There's a lot of companies that are going to do good and are going to make wonderful things, and there are open source things going on in the synthetic biology world, and they're wonderful and they're probably fundable. They're just not fundable by VCs. Our limited partners have certain expectations they've shared with us about the return on their money. And it's unlikely that an open source business plan is going to meet that goal. I mean, I can't even wave my hands and pretend. And so, absolutely, do the company, max out your credit card, write a grant, get an SBIR, and do it. It's just not all companies should be VC funded. That's all.
Let me actually add to that. So let's say that the biofab succeeds, or something succeeds. We create a community, we have a wonderful license, and we have open source biology. That doesn't mean a great deal, to tell you the truth, in terms of being able to pull it off the shelf. You do need people to indemnify it, to give quality control, and so forth. So let me amend what I said. Maybe we can go to Invitrogen, but they're selling the open source parts. Or maybe there's a new small company, graduate students that just finished from some school that want to start selling reagents that they've developed. And they can guarantee those. They can provide the quality assurance around those. So those are the kinds of models. Now, as David said, you're not going to get a huge return off of that. So it's a beautiful business, but for VCs, it's difficult to invest in those kinds of things.
So this is Rob Carlson up here. I just wanted to spin on that a little bit. VCs care about making money, right? And I don't think any of you are going to disagree with that. And that means, in general, they're investing in companies that are going to grow big. But within the U.S. economy, 99% of the employers are actually small companies. And those small companies employ 40% of the people in the country and pay about 45% of the total private payroll. So without the small companies, you don't get an actual economy. And I'd like then to ask if you guys have any strategies beyond what Tali said for expanding somehow the role of small companies or access to funding for small companies, or somehow building out the bioeconomy so that it looks like the rest of the U.S. economy. Or do you care?
Yeah, I think this gets back to what Jack said, because a lot of the venture capital goal was to take small groups of people, very talented people, and smaller amounts of money, tens of millions of dollars, build it into something that an investment banker would then sell for a lot more. And as Jack pointed out, there aren't that many investment bankers anymore, and that model is definitely broken. But I don't think that's actually something that we as people in systems biology have to be panicked about. Because in addition to being confident about our ideas in the future, we can be persistent about it and we can find money. Now, it's no accident that two of the flagship system biology companies, Amyris and Synthetic Genomics, both had people involved who knew how to raise money and get money non-dilutively. And so the important thing is to be persistent. If you don't know how to do something, that means you need to find the people that do, and you need to be open about finding new ways to raise money and to build value. and most importantly, focus on innovation. How are you creating something that's valuable? I think one of the great things, one of the great success stories of Silicon Valley is Google, and what was their motto? Don't be evil. Great thing about Amherst was they took it up a step higher. They said, hey, let's do something good. And then on top of it, the founders were very passionate about let's do something good for our next thing beyond malaria. Let's do something good with this new field, synthetic biology. Let's develop it and expand it. So I think you can afford, even though times are tough, I think it's a great time to be visionary and persistent. And it's good to be confident, but be flexible in your confidence to go out and work with go find other great people to work with. Or maybe you work with these larger companies. So one of the things that we believe in at CMEA Ventures is this whole concept of collaboration and mutual leverage. So for example, one of our companies, Codexis, which wasn't started as a synthetic biology company, although you can argue that gene shuffling is related to that, they're using tools of synthetic biology. And when they went into biofuels, they went and got a partner, a large company, Shell, and that's allowed Codexis to grow very dramatically and build their capabilities. So I think it's this whole idea of collaboration, mutual leverage, persistence, and flexibility.
Could I actually say something? We've got two tools in QB3 to address the problem you raised to starting small companies. One, we have what we call our garage, which is really a micro incubator, which allows us to rent out space down to 150 square feet, which is about 5 times less than the average real estate people will do. And so that allows faculty to start with very small burn rates. And the second thing, with the advice of Jack, we're actually creating our own venture fund, which we're able to fund at a very much smaller level than the average venture fund comes in at. And therefore, hopefully by getting equity in the companies that we help create from QB3, we'll get a replenishing fund, a little bit like the Dishpendi Fund at MIT. So these are tools we've tried to bring in to address exactly the point you raised. Other questions? Yes.
Could you comment on what terms you would offer people working in a biofab?
So the biofab would be a public effort. It wouldn't be a private. It would be a what's the best way to the best analogy to the biofab? What's that? GenBank. Potentially, I think but the biofab would be an engineering effort funded by either government sources or by private philanthropy. It sort of gets, and the goal there is to build some of the basic tools that would lend themselves to Rob's question. What do a number of these smaller companies need in order to bootstrap and perhaps get to the proof of concept and then come to some of these to some of us? Yes.
May I ask a question of the group here? Talking to Drew recently, I was impressed by the fact that synthetic biology can be used for all sorts of things you might not have first expected. So Drew was saying at a meeting he was at, someone was there from the perfume industry and pointed out perhaps you could use synthetic biology to make the source materials for perfume. So I want to ask the panel if they've heard of any unusual uses of synthetic biology that might be the basis of new companies.
I know of a very well-known company that talked about applications, exactly that, perfume, early on. So I think one of the great things about synthetic biology is you can prototype very rapidly and you can build many, many different variations. So if you can figure out a way to do the screen or the selection, you can begin to do experiments in a totally revolutionary way. So rather than doing experiments, I used to do northern blots, which used to actually measure one transcript level at a time. Now you just measure all of them, same amount of time. So that's the same thing that we're going to see with the increases in synthesis and ability to confirm via sequencing. So I think even one of the things in the biofab or in the BioBricks I think is good is the interoperability of being able to transfer things. But with the increases in synthesis, I think it might be possible for everybody just to order everything, and if, whether we order it from Invitrogen or any of these other companies, I think that actually could be good. That's a good collaborative thing, being a little bit older and having made stuff. And if you can't afford to, so I think it's also good to academically come up with new tools to be able to synthesize things as well.
More questions? Yes?
What application area that we haven't heard anything about is medicine or medical kind of application? Is it that that's filed under biotech, or would you say that that has something to do with the, you know, there's some commercial ventures, in synthetic biology related to this? How would you file that?
So I spent about 15 years selling into pharma. And I went to my first biofuels bioprocessing meeting about 2.5 years ago, and there were 3 more zeros behind everything. It's a very big difference, and there's no FDA. So you're going to see things, the chemical industry, the fuel industries, there's going to be applications as sensors, things that aren't regulated. You're going to do the low-hanging fruit first. Nobody's exactly, sure how the engineered bug is going to be used, how the FDA is going to treat that. So certainly medical applications are coming. Arguably creating drugs is a medical application. I think that you're seeing chemicals and fuels simply because the market is so big and so chaotic, and so that anybody who can make something can probably sell it.
So if you look at the reason why VCs are talking clean tech, why you sort of have to position everything in the context of alternative energy or petrochemical or biochemicals, is because of what David just said. We're talking about biotech, maybe a $250 billion industry. The petrochemical industry is in the trillions, and we need to replace that stuff. So the point why synthetic biology is so important for that space is you're talking about huge amounts of product with very low margins. So we have to do it better, faster, cheaper. So certainly there are opportunities in biopharmaceuticals, but synthetic biology is really well applied for the biochemical industry.
Yeah, hi. I am following on that. I was hoping, so government funding, government grants has come up a number of times. I was wondering if you guys could discuss quickly not only that, but government contracts and how that maybe would shape, product selection. I'm thinking about the $5.6 billion for BioShield, President's Malaria Initiative, PEPFAR. Has that influenced at all business decisions?
The DOE's 30 by 30 effort was driving a lot of the biofuel investment. I don't know if there are any governments with money anymore, so whether those incentives will remain, we'll see.
Yeah, you can go too far on government funding. There are quite a number of very promising startups that end up becoming SBIR factories, where all they do is make SBIR grants work. And I think you need to catch yourself before you get to that point.
But non-equity funding is really good. The same way the Gates Foundation really helped Amyris, and we've actually helped some of our companies when we start. If they're not thinking about grants, we say think about grants. If they don't have time to write the grant, we hire a grant writer or help them find a grant writer to write a grant. Absolutely, SBIRs are the lifeblood of 3 guys in a lab bench in Reg's garage. And that helps a lot. The trick is never to be distracted from your core focus. And George is right. The SBIR factory is a death knell. But we made the Megabase. That was a $32 million grant from the National Institutes and NIST, right? And so we couldn't have built that instrument without that grant. But along the way, we reduced the cost of sequencing in order of magnitude. So absolutely, they're not mutually exclusive at all.
One of the things in the biotech area, it's very clear there are clusters of innovation. Everyone knows what they are. Does any of the panelists see where clusters of synthetic biology are creating? Where going? And what might Hong Kong do to become a center of a cluster in synthetic biology? Any thoughts?